Good afternoon and welcome to your class, ATC 111. So, good afternoon, Teresa. So, we are now actually on, well, we,
Good afternoon, ma'am.
so, I think na maka last time, no?
Naa, ma'am.
Yeah, so, we still need to finish up, although I'm expecting na some of you have already gone through the genetic code, no? And so we will just continue a little bit and then we'll do the back cross method, you know. And hopefully I will just run through kasi we're really running out of time. So I guess nag-start ay nag-end somewhere in translation, no? So wait. Okay, so when we say again, translation, this is the last part na on genetic coding or making proteins. So sa prokaryotes, the translation is initiated with initiation complex, no, with an initiator tRNA and then N4-meal methionine and the small ribosomal subunit and mRNA strand. And then the ribosome binding sequence ng mRNA is complementary part ng RNA. And this is shown in a short while with this figure, no? So I will not explain further kasi anyway, this is really um, very technical and very deep already compared sa ating practical science. So, but then I just to show you how does this translation takes place. Yung kanina na mention na initiation complex and so on. And then elongation ng translation involving amino acids, addition, and so on. So this is shown here on this figure. Itong addition ng amino acids, then the how does this peptide bond is formed, and then the different amino and terminals from amino acid 1 hanggang 7, no? And this one with this cycle already, no, for the starting with the, although since this is a cycle, so there's no actually start or end, so it can start anywhere and end anywhere. So it's first matching tRNA anticodon with mRNA codon, and then peptide bond formation and translocation ng ribosome. So these figures are just on the cellular and genetic level already. So in translation also, there are fewer tRNAs than codons. So we have the so-called Wubel pairing, which allows less stringent pairing between these three base and so on. And this will allow fewer tRNAs to accommodate all the codons. Then elongation continues and the ribosome encounters a stop codon. So kanina, the stop codon are recognized by release factors which release the polypeptide from the ribosome. So it is shown in here, the release factor and the polypeptide chains, then they dissociate, no? So, and in eukaryotes, the translation also occur in the ribosome or in the cytoplasm or an ribosomes of the uh, RER, and then signal sequences begins with polypeptide sequence and bind to the SRP, and then this recognized by the RER receptor proteins. And then the signal sequence holds ribosome on the RER, and then polypeptide is synthesized and it passes through a pore or in the endoplasmic reticulum. It's as shown here. So from this SRP, signal recognition particle, and then it binds to peptide, and then here in the RER. So yun lang, no? And The most important is sometimes happening is the mutation, or it's called altered genes. So we have the so-called point mutation, which alter a single base, or base substitution mutation, substitute base for another. So yung base niya ang may substitute. This is the missense mutation. We also have the nonsense mutation, or the create stop codon, and the frame shift mutation caused by insertion or deletion of a single base. As shown here, this is a silent mutation wherein you have the sense template and protein na may ganito. And this is missense mutation, and this is nonsense mutation. So may kaibahan siya. And triplet repeat expansion mutation can involve sequence ng 3 DNA nucleotides that are repeated many times. That's why triplet repeats expansion. And these are associated with human genetic diseases. So kapag most of the human genetic diseases,
Good afternoon din.
good afternoon, Lovely, and also Chris, Kenneth, and again, Teresa. So, in continuation to our last topic, we just have started, no, yung translation. And before, after, in the end, ang last part ng proteins making, 'di ba, is yung stop codons, which is translation, and then magkakaroon ng stop codon. So we are now dealing about mutation, no? Paano pag may mutation, paano nangyari? So it's just an altered genes, and there are several ways, yung kanina missense, or yung kanina I can show you. Adi siya nga. So okay, here, the silent mutation, missense, and nonsense mutation. So yung nonsense, nagkakreate ng stop codon, and frame shift mutation, merong insertion or deletion ng single base. So, yung missense mutation is nagsasubstitute ang bases, yung mga A, G, T, yung ganon, as shown here. Okay, so I'll just have to be fast, ha, kasi anyway, you don't have to memorize all of this. All you need is just to be exposed, no, and not really to be at the deeper level dahil hindi na naman kayo magiging mag-genetics, or we don't know, maybe someday you will be venturing genetics, no, or plant breeding, or biotechnology, or um, somewhere breeding, no. So we are now here. Yeah, most of this triplet repeat expansion mutation are causing human genetic diseases, causing the abnormal allele diseases. Pag nagre-repeat siya, so yung normal allele uh, does not work well. So in the chromosomal mutations also merong change ng structure ng chromosome caused by deletion of the part of chromosome, duplication, and inversion and translocation. So this is also shown here. Merong deletion or nagduduplicate, or na-invert siya, nag-interchange in reciprocal translocation. Nagta-translocate siya, nag-iiba ng location. So too much of this genetic change called mutation can also be harmful to the individual, or to the human being, or to the plants maybe, or to the animals. These are caused, genetic variation then occurs, which are caused by mutation, yung kanina missense, or nonsense or necessary. This is also sometimes necessary for evolutionary change ng species. So, like the virus, ang bilis mag-mutate, diba? So, I guess yun lang about our topics on genetic coding. So, I have actually added some photos here Wala din na add ang katong circle. So, photos here for you to find out eto ha. Kasi I just have added your exam a while ago. So if you will look into your classwork, meron nag-post ako ng midterm and final. I think nandito sa midterm ninyo yung that you need to answer the genetic coding, diba? Simple na kaayo. So you just have to add the letter here, here, and there. So, kasi yung mga different amino acids are already, ang letter combination na lang ang i-fill in the blanks ninyo. So, if you have questions, then you can just clarify later. So, our next topic will be about yeah, we're still on module 2 pa pala. Backcross method. So, ano itong backcross method? Any one of you who have remember kung ano itong backcross method? So, backcross method is actually yung dapat anyway, try natin. Ay, sorry. So, this backcross method actually is this is for plants, and this also can be applied in animals like but for plants, for animals kasi like human beings like sa atin no, hindi pwedeng within ka mag-anak, magbi-breed or uh, kakasal tayo within sa ating mga relatives kasi we believe that um, it's not good for breeding. So, nakadungog damba mo nga kanang ano, kanang naay mag-ig-agaw ng binyo or mag-igsuon?
Yes, ma'am.
Sa TV.
Mag-ig-agaw.
And
Samal to, ma'am. Taga-Samal.
talaga, and then how did the community react on this, or how would you react on that?
Ang ilang kuan, ma'am, kay maaman to sa mga ritual gibuhat, ma'am, baligi kuan daw gipanglabay daw tong mga gamit nila, tas gipang sunog daw niya, dili kanang wala na nila giila og agaw ay marag di na nila ilahon ang kaning babae nga pag umangkon, ang kaning lalaki nga pag umangkon. Ana lang man, ma'am.
But they don't know that it's been a long practice in the royal family, no? Although di man ta royal, wala man tayo royalty dere, but way before in the Philippines, naapot tayo mga lakan o lakandula, diba? Kada mga hari kunuhay o mga prinsesa. But in the, like in UK, in Dubai, and even in Thailand, no? They just marry within their cousin, there's in their circle, no? Mga ig-agaw na sila ano, nagminyo-minyo. So, but lovely, unsay, nagkaanak sila, love.
Yes, ma'am.
And then, unsay lang anak, okay ra. Walay defense.
Okay lang man ilang anak, ma'am.
Maayo eh. So, actually, kasi we believe, scientifically, we believe that that is actually a fiction, na scientific fiction, kasi there are also claims na actually mas madami pang although yeah, that happens, no, kung like when you only like for animals, 'di ba, kung halimbawa mag-breed ka sa anak o sa iyahang inahan, sa parents, and kung mag-breed ka within sa ilahang family, close family, so we thought na lain ang effect no sa animals, or if or napa siya adverse effect. So just like humans, no, we believe na scientifically wala mura ganig walay hybrids like ikaw no, syempre kita normally kita ma-hybrids na ta kaya na cross ta between different genes sa ato ang parents, but so that is to improve, so always improve through breeding, no, and cross pollination for plants. But in animals, that's why gusto nato lahi na pud nga family, dili ta mo balik sa sisters, dili ta mo balik sa iyahang parents nga ano, ipabreed nato siya sa ubang breeds para ma-improve ang atong breeds, no? That's our principle. But then they are also saying that mas in terms of like we say mga mutation or kanang mga genetic defects, they also say science also claims that mas madami pang defect ang mafo-form kung halimbawa dili compatible ang mga parents, no, when they cross, than compared sa within family lang i-breed nimo. It's either animals or plants I'm talking about, ha. So mas konti lang actually based on data ang mga genetic defects compared sa mga crosses or hybrids, no? That's that. And it makes sense kasi we don't know kasi ang compatibility ng bawat species, no? So, kumpara sa i-breed nimo ang within the same family, the same genetic na ano data. So, in a small circle, less lang po talaga yung genetic disorder. But then again, going back to Backross method, that means this is the method of breeding wherein the hybrid and the progenies in subsequent generations are repeatedly backcrossed to one of the parents. So yung mga anak, ibalik na po breed sa parents, no? As a result, the genotype of the backcross progeny becomes increasingly similar to the recurrent parent para makuha na, para mas similar umubalik kasi like ang parents nimo lahi, and then ikaw nga anak hybrid na ka, so gusto na pud nimo nga katong, so i-breed ni po, halimbawa we're talking about baka ha or baboy, i-breed na po nimo balik ang anak sa parents kasi gusto ka katong sa isa ka parent nimo na mga genetic characters ang mugawas. So this is way of parang making the genes pure or going back to the original genetic parents. So they only do this kung naa sila specific character nga naa sa parent sa breed, no? And the objective is to improve one or two specific defects of a high-yielding variety. For example, in plants, I have a project before known as abaca hybrids, no? Abaca hybrids trial in the Philippines. So I'm one of the project leader there, and it's actually a virus-resistant abaca. Kasi yung abaca before, don't you know that abaca is Musa textilis is only indigent in the Philippines, all over the world. But now it is also found in somewhere in somewhere in Latin or South America, Colombia, I think. No, it's sa nga country nag-start og U sa South America. Nakalimot ko, sorry. But if ma remember na ako, then I will mention it again. So actually, and they said that historically, it comes from the Philippines, no? So, and in fact, when ang Pilipinas ay masyadong mayaman pa, so one of our major export is abaca fiber. So now we are part of the purpose of that study is kasi may havoc nung ano mga decades before. So, na wipe out yung abaca natin because of virus. And now, they're breeding virus-resistant abaca hybrid. So, nakakita sila ng hybrid ng ang breed na resistant sa virus and then binabackcross nila kasi yung musa species na yun ay sa saging, hindi sa abaca. So, nag-breed sila ng abaca breed, ng the best na breed ng abaca and then yung abaca na yung nusa or saging na breed na resistant sa virus and ang ginamit ay backcross para makuha yung maibalik yung resistance na yon. At the same time, pang fiber din siya. So, it's just an example. So, there are prerequisites for back cross breeding. So, dapat suitable recurrent parent which lacks in one or two characteristic, and a suitable donor parent must also be available. The character to be transferred must have high heritability and preferably determined by one or two genes, and sufficient number of backcross should be made so that the genotype recurrent parent is recovered in full. So ito yung mga qualifications before you can use it for backcross breeding. So ang applications naman nito, it's used for transfer ng disease resistance as I have explained a while ago from one variety to another. And it's also used for the transfer ng ibang characteristic. No? Now, first part is intervarietal transfer ng simply inherited characters such as disease resistance, seed coat color, and intervarietal transfer ng quantitative characters such as plant height, seed size, and seed shape, and interspecific transfer ng simply inherited characters such as disease resistance from related species to cultivated species. For example, yung resistance ng black arm disease in cotton from wild tetraploid species into G. hirstulum, Gossypum hirsutum. And this is cotton. And transfer of cytoplasm, no, employing transfer to transfer male sterility. So, bakit naman gusto nila para mas more sterile? Female parent will be having the sterile cytoplasm and the recurrent parent will be used as a male parent. For example, the Sesamum malabariucum and indicum. This is the female parent and the recurrent parent. And there are actually a lot of uses, including the transgressive segregation. So in this backcross method, this is used to modify to produce transgressive segregans. So the F1 or the first filial generation will be backcross to the recurrent parent for 2 to 3 times para makuha mo yung transgressive segregans. So, and production ng isogenic lines. So malalaman natin later what are this, and germplasm conversion. So example are production of photoinsensitive line from photosensitive germplasm through backcrossing, and this was done in the case also in sorghum or yung popularly known as conversion program. So there are 7 uses and ano yung procedure. So I will not discuss into detail, and if you are very interested, and you can just go back to this link later. So the plan ng backcross method would depend whether the gene being transferred is recessive or dominant. So the plan for transfer of a dominant gene is simpler than of recessive gene. Siyempre, kapag i-cross mo yung dominant gene na gusto mo, yun talaga ang lalabas, di ba? So, palpak na dyan kaayo kung dili gid mo gawas ang dominant. Like, for example, no, kita mga Pinoy, ganahan yung tagputi, no, kasi pare sa to ang puti ang gwapa. But it's not really, no? It's just psychological. So, halimbawa, putiin mo mama o papa, and then dominant is puti, but then ikaw, sa tanan nimo mag-isuon, puti sila, ikaw ra iton, di ba? Pagkamalas na lang nimo. That really happens sometimes. Although recessive ang black, but then supposedly dapat puti pud ka, but sometimes that would happen. So in the first year, non-recurrent and the recurrent, the parent B and the parent A, for example, yung parent B or non-recurrent is have the resistance to RAS. Yung isang parent is susceptible to RAS. So you want this resistance uh, to take this resistance. So you just um, cross them, no? So you have here the both resistant and recessive, no? And in the first back cross product, so you will have the first filler generation, may combination na resistant to rust and susceptible to rust. And then yung F1 gagamitin mo na naman to combine itong RR to combine with the susceptible rust kasi gusto nimo ang pagka-resistant ani madala aning parent A. So, then you're gonna have backcross 2 effect, then 3, 4, and 5. No? And backcross up to 6 to 7th generation. Then after 7th generation, yung backcross rust resistant lines will be self-pollinated na. No? And then will be harvested on a single plant basis. On the 8th season, the individual plant progenies will be grown and the homozygote plants with a resistance and resembling parent A will be selected again. And in the 9th season, yield trials. On the 10th season, mag-multiply and distribute ng seed. So so this is the first steps, no? So in the first season, hybridization or crossing yung mga kanina. And the second season, yung F-line backcross to recurrent parent A. And third season, again, like what is shown in 4th, 5th to 8th, 9th, 10th, and 11th, 12th, no? So, yun yung process. So, there are actually different types ng backcross method, no? If you want to transfer the recessive gene, so ito yung another method to do it. So in the first season, again, i-cross mo sila, hybridization. And second season, third season, backcross 1, and then backcross 2, 3. So in the 6th, 7th, and 8th season, so instead, itong backcross 3 and F1, no? Ito yung kaibahan niya. For magba-backcross F1 ka and backcross 2 F2 na po hanggang balik ka na naman dito sa 8th season. Backcross 3 na siya, but you go back to the F1 again, the first, and so on. So sa method siya nagkakaiba. No? As I have said, I will not dwell so much kasi we're running out of time. So again, we have different procedures na nakita natin. Yung una, if it is ikalawa is if it's recessive gene. Ito if it is dominant, if it is the dominant genes. So I hope you understand what I am talking about. And the merits of this or yung advantage nga is that yung new variety is nearly identical na with the recurrent parent, yung gusto mong ibalik na characters ng parent except for the genes transferred. And the outcome is known already, no, and it can be reproduced anytime in the future. And it also not necessary to test the variety developed by the backcross method. No? So hindi na kailangan siyang i-test. And this may save up to 5 years, considerable expense. So mind you, breeding is will take 10 to 25 years, and with and yun nga, for backcross method, you save 5 years. And also the backcross program is not dependent upon environment except for that selection ng character under transfer. And therefore, pwede kang off-season nurseries and greenhouses, no, for 2 to 3 generation is here. And again, reduce the time, and much smaller population are needed in the backcross method in the case ng pedigree. And defects such as susceptibility diseases and so on can be removed without affecting performance adaptability. So variety is often preferred by the farmers. And this is the only method for inter-specific gene transfer. However, it can also be modified so that transgressive segregation may occur quantitative characters. Yung disadvantage or demerits naman, yung new variety cannot be superior to the recurrent kasi nga binabalik mo yung original na character ng parent. So it's not actually a hybrid, na hybrid siya pero it's not superior. So undesirable genes closely linked with gene being transferred may also be transmitted to the new variety. And hybridization has to be done for each backcross, which is difficult, time-taking, and costly. And by the time the backcross is over, the recurrent parent may have been replaced by other varieties superior in yielding abilities and other characteristics. So ganon. So in terms of number of plants, so only few lang, no, ang kailangan, like 10 seeds for each backcross generation and so on. So I cannot explain everything because of the time, but this is very self-explanatory na, and how to select the character being transferred, number ng backcrosses to be made. These are very specific na methods, and there are also modification of the backcross method such as production ng F2 and F3 generation if you want to use it, and backcross pedigree, the pedigree method wherein the hybrid is backcross 1-2 times to the original or recurrent parent. And the application of backcross method to cross-pollination. So may application siya, no, which is in the almost similar to sa pollinated crops. No? So I guess also here mayroong comparison ang pedigree and backcross method. So, and also it's used in multi-line varieties. So I hope you will have time to read this and the merits of multi-line varieties and the merits. And so far, yung achievements natin in terms of multi-line variety is very useful in the control of diseases like rust in the wheat, no? Or TA's tubum. So these are the different varieties of wheat. So usually, ang gamit talaga ng backcross method ay for disease resistance. Yeah. Yun lang ang parang main gamit bakit we do backcross method. So in the animal and plant breeding, we have learned the different types of breeding system for those animals and plants. So, and backcross method is one of the method in breeding. But then in breeding, usually para we breed in order to, and also in order to, to improve our varieties. So if you want to improve varieties, you don't want to use the backcross method kasi pabalik siya. Okay, so do you have any question regarding backcross methods, so we can proceed with the overview ng plant genomics? Jobert, nabot lagi kayo ka sa mati ka.
Ma'am?
Ay, ikaw to ay nag-ingon diba na mag-work ka sa mati ba to?
Si Jules man to ma'am, siya digo sa ata, Jules.
Sorry, Jobert, Jules dahil. Sorry. Asa na ka karon, Jobert?
Sa trabaho ma'am.
Nag-work po ka, nag-multitask ka darang dapita. So,
Yes ma'am.
we are just less than 5 minutes now. So, I'll just introduce you and I hope you will read in continuation kasi 'di ba we have much time but then we are only meeting for virtually for 45 minutes. So, ano itong plant genomics? So, these are recent technological advancement, no? And these have substantially expanded our ability to analyze and understand plant genetics or genes to reduce gap existing between genotype or yung ating characteristic in genes and phenotype yung ating physical characteristic. So this field of genomics will allow scientists to analyze thousands of genes in parallel to understand the genetic architecture ng plant genomes and also to isolate genes responsible for mutations. So we are actually hoping that in animals naman and humans that kasi sabi nila yung mga pinakamatandang tao na nag-live up to more than 100 years are actually caused by altered genes or mutation. So there are also, but we are talking plant genomics, no, but in animals. And so they are wondering if they are trying to study na kaning mga genes responsible for mutation to apply this for living forever. Grabe naman, no? Or in plants naman, para to preserve our plants, no? So this is actually a review on, and also the genes of particular plants can now be sequenced, no? Pwede na nato ma-enumerate tanang genes na nasa tao because we have human genomics and also most of the plants already, and I have here figures. And also this review already discusses ways to extract biological meaning of the DNA data, and this this also provides insights to analysis of other organisms. So over the years, ito yung mga na-clear genome size of different species. So in terms of sizes, these are the following plants, no? The wheat, garden pea, asparagus, and so on. So try to look at this, and we are actually having Arabidopsis, no? Ito yung model plants natin, but we also have model organisms like Drosophila and Saccharomyces cerevisiae, yung bacteria, and Caenobartis. So I hope you will read this in advance, and I'm just going to go back here because then we are less than a minute. So if you have questions also, you can also ask through the group chat and try to answer na your mga requirements. So we have 1 month more to go. So ito yung map na ng plant genetics. The physical genetic map, no, and this is in 1 centimeter, 10 centimeter ito. So actually, these are just, and this is the whole genome sequencing, no, clone by clone approach. So this is how they do the shotgun approach. Yan. So ito yung mga steps. So I will not, so these are the gene complement ng Arabidopsis thaliana. So ito yung mga model plants. No? And sa humans natin, yung ginagamit nating model ay yung mga mice or daga. Kasi parang, parang against siya sa human rights or, you know, yung morality kapag ginamit natin sa laboratory ang mismong tao. So we use mice as for humans na studies. So ito yung for plants naman ang Arabidopsis thaliana. So ito yung feature with the different chromosomes, no, may 5 types of chromosome with various features from DNA molecules to